Today we're going to talk about angiosperm life cycles, or flowering plant life cycles. As in past videos, I like to put the dominant stage on top, which is going to be the sporophyte. So let's go ahead and start with the sporophyte. And down below we're going to put the gametophyte. Now the sporophyte is going to produce spores, and the gametophyte is going to produce gametes. That's our first layer. Let's go ahead and add some additional information to this. And we're going to produce two types of spores, just like we did with gymnosperms. We're going to produce a megaspore and a microspore, which would be the male spore. Two types of gametophytes. We're going to have a female gametophyte, also known as the embryo sac. Now the embryo sac consists of a couple different types of cells. So I have another video out called the carpal video, and that is going to talk a whole lot more about the embryo sac, so make sure you watch that. Now the male gametophyte is still going to be the pollen grain. And then gametes, the male gametophyte is going to produce, again, two non-flagellated sperm. And the female gametophyte, or the embryo sac, is going to produce eggs. Now let's go ahead and connect these things up. The sporophyte produces a megaspore and a microspore. Two different size spores. Again, this process is going to be referred to as heterospory. Two different sized spores. And the megaspore is going to grow inside the ovary. And the microspore is going to grow in the anther. And the megaspore is going to further differentiate and develop into the embryo sac, and the microspore is going to further differentiate and develop into the pollen grain. And again, the spores stay put. So they stay on either the anther or the ovary where they're located. The pollen grain is going to produce two non-flagellated sperm, the same as with gymnosperms. And the female gametophyte is going to go ahead and produce eggs. Now the pollen is going to get dispersed in two different ways here. Some angiosperms stick with the wing pollination. Others are going to take a more efficient route and recruit the animals. So we'll write recruit animals. So who are we going to recruit? We're going to recruit the insects. We're going to recruit the birds. And believe it or not, we're going to recruit the bats. So unlike Hollywood's, how Hollywood portrays the bats, they're really good guys. They help pollinate a variety of crops and a variety of plants. And they also, one of their main dietary foods is insects. So they reduce the number of insects by huge amounts every night when they come out. And that's a good thing because some insects carry disease. So that helps reduce the incidence of disease. If we lose our bats, the incidence of disease is going to go up because the insect population is going to go up. So these guys are really important creatures. Let's see here. And our two non-flagellated sperm are going to come together with our eggs. Again, the sperm are going to come from a different plant because we want to maximize genetic diversity here. Now let's go ahead and write down our ploidy, how many sets of chromosomes we have here. The sporophyte is going to contain two sets, so 2N. Everybody else is going to be N, a single set of chromosomes. And using that information, we can figure out how we get from here to here. How do we reduce the number of chromosomes in half? That's going to be meiosis. N to N is going to be mitosis. N to N again, mitosis. And here, what we're going to have, N plus N to 2N, that's going to be fertilization. But here we're going to have a double fertilization event. So I'm going to go right ahead and write that down. A double fertilization event. Now what does that mean? That means one sperm fertilizes the egg and becomes the next sporophyte, and the other sperm fertilizes a central cell. A central cell. That's going to be a cell that's located in the embryo sac. And this central cell is actually going to be 2N. It's actually going to be diploid. And once that sperm, which is N, fertilizes that central cell, which is 2N, we're going to get a new substance being produced here called endosperm. And N plus 2N is going to give us 3N. So our endosperm is going to be triploid or triploid. And the endosperm is not like a sperm. The endosperm is a food source. It is food for the developing embryo. Now once fertilization occurs, again, just like gymnosperms, we're going to get seed development. And the seeds are going to consist of 3 parts: an embryo, a food source, which is going to be our endosperm that we just created here, and also we're going to get a seed coat. And the seed coat is going to provide protection to the seed. Now our seeds here could be dispersed by the wind, but oftentimes our seeds actually get dispersed by animals eating that seed. They're going to eat a fruit that contains that seed, and not all the seeds are going to get chewed up. Some of them just get swallowed whole. And this seed coat actually prevents those seeds from being digested. You've probably seen this before when you've eaten corn. Corn kernels are actually seeds. And if you don't chew them up thoroughly, they come out just like they went in, intact. And that's what happens here, and these animals will then walk around the environment, and when they go to the bathroom, the seed comes out plus a little bit of fertilizer, and that seed is set to go and grow into the next generation. So the angiosperms really take advantage of the animals here and get those animals to very efficiently pollinate and disperse their seeds for them. And that's going to be it for angiosperm life cycles.